Yeah, so related to the earlier question, many scientists nowadays, because of the single-cell RNA sequencing technology are becoming available, they are starting to do more single-cell analysis of the tumors. And basically, previously, when we have the resected tumor, you just go directly to a bulk RNA-seq. And now, if we can generate a single-cell suspension, you can do single-cell RNA-seq. You can also do single-cell attack-seq that we discussed at the end of the module 2. And for example, in the early days for glioblastoma, so this is a stage 4 brain tumor. If you just look at pathology, early days, people thought, okay, maybe there are only 2 subtypes, right? There's astrocytoma and oligodendral glioma. So these are 2 subtypes. But once you do TCGA tumor expression profile, looking at both states, so these are all the brain tumors. GBM is just a more severe type. Low-grade glioma is like an earlier stage, I guess. If you look at these, based on the gene expression, it was determined there were like 4 subtypes, roughly. There's a classical, mesenchymal, neural, and proneural subtypes. And so clearly, actually, these 4 subtypes are also associated with different patient outcome. Interestingly, recently there is a study. Sorry, I forgot to add a citation. I'll do that. It's a study from Mario Suva's lab last year. They did single-cell RNA-seq of the tumor. And interestingly, when they saw this is, there are 4 different cancer cell clusters. These 4 different subtypes really drive the tumor to have different heterogeneity. You can imagine heterogeneity could be different patient. You know, this patient have the breast or glioblastoma, and another patient the glioblastoma being different. But interestingly, in this situation, they see actually there are 4 different subtypes, and every patient has a little bit of those cells. And if they are the one, say the neuroprogenitor cell-like, they just have more of this population and less of the other, but everybody has a little bit of the other cells as well. And so there are very big plasticity between the 4 states. And also some of them are also driven by different gene expression or mutation status. Usually, I think in this case, is gene expression patterns driven those tumors to be quite different. Although, I guess because of gene expression, it could also have plasticity to move from one to the other. Maybe with some drug treatment, you know, one group will be less, and they will shift to another group. That's how potentially cancer cell can develop resistance. And so, basically what people observe in this TCGA subtypes here is just a matter of whether the patient has more of this cells or these cells or these cells or these cells. That's, you know, how you go to 4 different subtypes. But actually every patient in their tumors have some populations of all 4. It's just different proportions, right? So you can see within the same tumor, you have more heterogeneity. So of course, these are kind of expensive assays. And so many scientists nowadays are collecting patient samples and now doing single-cell RNA-seq of the tumors. And I think we still have a lot more we can learn. And this is only on the cancer cells. 2 lectures from now, we'll also talk about the whole tumor microenvironment. What other cells are there that you can learn from? So in 2000, Hanahan and Weinberg wrote this Hallmark of Cancer paper where they show, you know, what are the major drivers of cancer. So first you say, okay, you need to have sustained proliferate signal. So basically the gas pedal needs to be on all the time, and evading tumor growth suppressors. So the brake is taken out from the car, right? So gas pedal keeps going and the brake is gone. And then activating invasion and metastasis. So basically this tumor will also metastasize to other tissues or organs. And also enabling replicative immortality. So our normal cell after a certain cell cycle will die, but in cancer cells, they don't really care. They can keep replicating. That's how you can grow cell lines in Petri dish forever. Also, they can induce angiogenesis, kind of help the blood vessels to grow into the tumor, to supply the tumor with oxygen, with energy, and also resisting cell death. They don't die anymore, right? So these are, you know, what they discovered in 2000 as the main hallmarks of cancer. But in 2011, they had new hallmarks, and the previous ones are still there. But then they say, turns out every time you have tumor, it's because they evaded immune. And also there are some certain inflammation in the tumor, which could be viral infection or whatever caused the inflammation. Those actually are causing tumor growth. And also, whenever you have the tumor, that the genome is not very stable. It's like a viral, basically with every DNA replication, new mutations can evolve from the same tumor. And so the genome is kind of more dynamic. And if you treat the cell with drug, additional mutations can become resistant to the drug, right? And finally, the tumor has very, very different energy metabolism, as we saw in the DNA methylation IDH mutation. They are also energy metabolism differences. And because of this, in each type, there are more drugs that are being developed to target them. And so in the next lecture, we're going to tell you what people are doing in terms of treating tumors driven by different mechanisms with a different drug. That's real precision medicine, right? So that's it for today. Sorry for the delay. Hope everybody stay safe. Thank you.
Yeah, thank you.
Bye.